Okay, so let's talk a little bit about gene therapy. Give you a little background information, some history, and some process, and then I'm going to talk real quickly about one case study that we're going to spend a little time with, where you each are going to get an opportunity to kind of look at the various stakeholders in this particular research in gene therapy and sort of discuss whether or not you think that this is something good or ethical or unethical, if we will. So, a little bit about gene therapy. So, gene therapy is basically the use of DNA as a pharmaceutical agent to treat a disease. It gets its name from the idea that DNA can be used as a supplement to alter genes with an individual cell as a therapy for the ability to treat that disease. The most common form of gene therapy that we use today involves using DNA that encodes a functional therapeutic gene in order to replace a mutated gene within the DNA strand, within the nuclear DNA of the individual. In gene therapy, what we do is we take the DNA and we encode that therapeutic gene, which then turns around through the process of transcription and translation, translates that into a therapeutic protein. And so that therapeutic protein, which is the actual item that's going to work and do the job, that therapeutic protein is going to be packaged in what we call a vector. And usually vectors are a virus, believe it or not. We use viral components like a protein capsule, and we place the DNA inside that protein capsule like a virus, and we inject the individual with that virus, because essentially viruses are non-living organisms that are capsules, as we talked about in the vaccination unit. They're capsules with a strand of DNA or RNA inside. So we use them as the vectors in order to inject the target DNA that we want into a cell. So we use that vector and we inject it into the cell. We use that virus and put it in the cell. And once inside, the DNA becomes expressed by the cell's mechanisms of, again, translation and transcription, resulting in that therapeutic protein. And that protein will then treat the patient's disease or the symptoms of the disease, as you saw earlier. So let's talk a little bit about the history of gene therapy. Early work in gene therapy began in about the 70s as researchers started sort of developing the idea that we could take this process of a vector. And so here's a nice diagram where you can see this is a viral vector that gets recepted by the cell, gets received by the cell, comes into the cell, and then walks through the process of that transcription, translation in the nucleus of the cell. So they developed that idea, and the first clinical trials for this actually began in the 90s. They began talking about it in the 70s and the 90s. We saw our first clinical trial. Gene therapy has had some major setbacks over the years. There were some deaths of some patients in 2 separate trials that occurred that, of course, resulted in a much tighter control of the approval and the reporting process. And as we learned in the previous unit, human research is already very challenging to pass. So you can imagine when they had these situations how much harder it became. So there is some current trials going on around the world, obviously, but one specifically we're going to use as a case study here is one that occurs at University of Florida, and it started in 2010. And basically what happened is the researchers at UF began enrolling patients for what's called a phase 1/2 gene therapy clinical trial. And what that basically means is that it's an early phase and a secondary phase, so they should get an idea of what, the injection process does it take, and then ultimately does it transcribe that protein. Scientists have incorporated the correct gene into be able to produce what's called an enzyme called GAA, and we basically take that GAA and we put it inside of what's called an adeno-associated virus. And adeno is what we use. It's a common recognized molecule, adenosine, if you've heard that before, like adenosine triphosphate for ATP, things like that. So it's an adeno-associated virus, so the body recognizes it as something that it wants to take. Because if you think about it, your body is geared toward trying to fight viruses. So this is a specific virus which already kind of exists in people, the adeno-associated virus. So it's something that the body is accustomed to, and so we make sure that it's recognizable. And during the clinical trial, we inject that into each of the patient's areas. What UF did is they injected it into the diaphragm of the patient, because that's a very sort of centralized location in the body. The intent was to infect the cells of what's called the Pompe disease. There's a disease out there, it's POMPE, and so they were using specific patients that have what's called the Pompe disease, and they wanted to see if they could infect them with the genetic machinery that they may not have had that makes them basically have this disease. So if it was successful, they would be able to basically increase the quality of life for the people with the disease, and they're not curing the disease, but they were alleviating symptoms of it so that they could live sort of a very symptom-free life, basically. So that is the essential background information on gene therapy, and then on this particular clinical trial. And then you go back to the gene therapy page and you'll see what you have to do next.